Dear students, today's topic is automation in microbiology. So all of you know that so many automated methods are emerging. So what is the scope of these automated methods will be dealt with in today's topic. What are the drawbacks of conventional method? The conventional methods may miss the organisms if there is a low microbial load. So therefore, there is a need of more sensitive methods so that we can pick up the organisms in culture. The conventional methods are slow and time consuming. Even if there is a certainty of growth, it will take 3 days. So, to some extent, the automated methods will reduce the turnaround time. The contamination is another problem in conventional methods. This will be obviated by standardized culture media in automated methods. Their poor reliability and poor reproducibility, these are another two concerns of conventional methods. So automation was introduced in 1970, where the semi-automated blood culture system was introduced. The first automated blood culture system was BACTEC 460 and based on radiometric detection. So here the radio level carbon 14 was used to release the carbon dioxide, and the radioactivity is detected by the sensors which are there in the machine. But always there was a problem, and activity of the radioactivity hazard was the concern. Then afterwards, in 1980, there was a development of continuous monitoring of blood culture system, CMBCS, and the examples are BACT Alert, BACTEC 9000 series, and Versatrix systems. Then continuous monitoring of blood culture system, what it will do, it will monitor CO2 production at every 10 to 15 minutes, and the detection of the growth is done. Then that was continuous monitoring blood culture system is a non-radiometric. So it may be using calorimetric or fluorometric mechanisms of detection. So the disposal of the waste after testing was not the concern. Then automated ID. Automated ID was introduced for identification of the organisms and was introduced just after the first automated blood culture system was introduced. So Vitek system is the example, and it was developed by NASA to test the astronauts from unusual organisms acquired during their space visit. It is a calorimetric system. The examples of Vitek 2, Phoenix, Microscan, Drake diagnostic systems, and these are using chromogenic mechanism or fluorogenic mechanism for detection of the growth. And also antimicrobial susceptibility sensitive testing is also possible, and same principle is utilized for its detection of antimicrobial resistance. So what are the challenges in adopting automated methods? Complexity of the microbiological specimens. Always the specimens are contaminated with the normal flora. So if there is a mixed growth, again there will be the interference of other organisms and it becomes difficult. Non-specific clinical manifestations is another concern. Then high cost of investment and consumables. Cost of machine as well as consumables is always concern, uh, especially in our country where there is a lack of the proper funds. Then what are the current systems in microbiology lab automated? So first is automated specimen processing, where there is automated loop is there, magnetic beads, boom applicator, then automated ID and AST system, identification and antimicrobial susceptibility system. So radiometric blood culture is there, then continuous monitoring of blood culture system is there, ID and AST for bacteria and yeast, then mycobacterial identification system. Another automated is molecular automation. Then total laboratory automation is the next one. Then proteomic based automated identification system, that is a MALDI TOF, which is the most recent method. for the identification of growth and also antimicrobial susceptibility testing. So now come to the automated specimen processing. Automated specimen processing. Here, the quality of sticking process is improved, contamination is reduced, reproducibility of the result is improved, reproducibility of the process is improved, and there is high yield of isolated colonies. The examples are automated loop, magnetic beads, and comb applicator. So photographs are given in the next slide. Now you can see this automated loop where the machine automatically inoculates the plates in a standardized manner. So this is the example of automated loop. And then magnetic beads. We can use the magnetic property for inoculation of the samples. And here the system automatically agitate, decap, recap the specimens followed by inoculation on whole agar plates or biplates as required for the specimen type. And these equipments are equipped with HEPA filters. Now this is the developed by Biomeric French company. You can see here the applicator, comb applicator. Before the application, it is a yellow color. Now it is ready for inoculation. We have to stick on the plate where there will be the red color light, and after successful procedure it will show the green color, so that application of the inoculum is successful. Then we can remove it. Standardized manner there will be inoculation. Now come to automated ID and AST systems which are available. So before that we'll come to the comparison of conventional versus automated. Suppose if you are going for blood culture in conventional methods, we are growing in glucose or bile broth. You can see here photograph of glucose broth, and these bottles are incubated for 7 days, and every alternate day we are doing subculture 1, 3, 5, 7 days on blood agar and McConkey's agar. Then after growth happens, we have to go for biochemical identification and antimicrobial susceptibility testing by doing the manual methods. The result is able to obtain within 3 to 11 days. So there is so much manual interference. Repeatedly we have to open the bottle and there are chances of contamination. Turnaround time is 3 to 11 days, which is much more. Now continuous monitoring blood culture systems. As soon as once the bottle is loaded, the signal is received usually within the 6 hours to 3 days, and we have to subculture on blood agar or McConkey's agar. The growth is obtained, and then from that growth we have to put for the automated ID and AST. If both automated continuous monitoring of blood culture system and automated ID AST is combined together, result is able to obtain within 42 hours or to about 5.5 days. So there is a decreased turnaround time. You can see the photographs of the consumable here, which is a bottle, which is commercially available. We have to spend for again for the consumables. So this is the equipment, which is one-time investment, where there is a panel, display panel, LCD display panel, where it will show the bottle, and if there is a growth, it will show the slot where their growth is obtained. We have to take out the bottle and subculture again for further processing of the sample for identification. Now, radiometric detection is now history. In 1970, it was introduced. So, where the carbon labeled, that radioactive carbon was used for labeling, the CO2 released will stimulate the sensor, and which will give the indication that there is a growth. So, but there was a less frequency of the detection, once or twice in a day. But if we have seen the continuous monitoring of blood culture system within 10 to 15, every 10 to 15 minutes, there was a detection. So again, radioactivity, radio level, so discarding was hazardous. So therefore, it has gone now. It has become the history, radiometric detection methods. Then continuous monitoring blood culture system. You can see BACT alert 3D. which utilize the calorimetric method, where because of the chemical reaction there is a change in the color, and we can detect the change in the color by the sensor. The machine will detect. Then BD Backtech, which is a fluorescent based detection system, that is a BD Backtech. And then Versatrek, that is a newer one, where the pressure change in the head of the bottle is detected, which will indicate the growth in that bottle. And all these above, these are non-radiometric. So there is a disposal. That disposal concern is not there. Then automated ID and AST system. So these are the pH-based reactions. The example is Phoenix panels, Vitek cards, which will utilize the pH-based mechanism for a detection. Then enzyme profile, microscan rapid panels, Vitek cards, Phoenix panels. So these are the commercially available equipments. Then carbon source utilization, where that principle is utilized in OmniLog. Then volatile and non-volatile compound detection. It is detected by Sherlock Microbial ID system. So there is a competition between these manufacturing companies to provide different mechanisms for ID and AST. So you can see this table where we can see various manufacturers, biomerics, so Vitek 2XL, Vitek 260, Vitek 2 compact, then again the capacity 60, 30 or 15, and Vitek 2. So these are the instruments, and this biomeric company utilizes calorimetric or enzymatic mechanisms for the detection. And the panels are gram negative, gram positive, Neisseria, Haemophilus, anaerobes, yeast. So these are the various panels we have to use for identification depending upon our preliminary testing on the after the growth. And incubation within the 8 hours, the result may come, and even antimicrobial susceptibility testing is also possible. Then another product is by the manufacturer Siemens. So this is the microscan walk away plus. So overnight panels are also there, rapid panels are there. So overnight panels, turbidometric detection of carbon source utilization and enzymatic activity. And then rapid panels, fluorometric detection, where preform enzymes are used. And all these panels are similar like biomeric, gram positive, gram negative, Neisseria, Haemophilus, anaerobes, yeast. So each will be having different cassettes for their utilization, and their consumables will also, the cost will be different. Then incubation 16 to 18 hours, and we can detect the antimicrobial susceptibility testing. Then come to BD diagnostics. BD Phoenix is the brand name of that machine, which will utilize calorimetric and fluorometric mechanisms. Here, the panels which are available: gram positive, gram negative, streptococci, yeast identification. 18 to 16 hours is the incubation period, and we can do the antimicrobial susceptibility testing also. Then Trek diagnostics, that instruments available is AIRS, which utilizes the fluorometric mechanism, and the panels available gram positive and gram negative. The detection incubation 5 to 18 hours, and even we can be used for antimicrobial susceptibility testing. Then biolog is there. The brand name is of that machine is OmniLog. So here the principle is reduction of tetrazolium violate. That principle is used for detection, and gram positive and gram negative organisms can be detected. 4 to 24 hours is the incubation of this, and it cannot be utilized for AST. It is only for detection. Then mini INC, which is the instrument name is Sherlock Microbial ID, which detects the cellular fatty acids by gas liquid chromatography. We can detect gram positive organisms, gram negative organisms, yeast, and incubation period is 24 hours. So these are the various instruments, but here one AST cannot be done. So last two, that is biolog and mini INC, AST cannot be done, only detection is done. Remaining others, we can go for AST also. So this is the spectrum of the commercially available instruments we have to pick up depending upon our requirement. We have to study before putting the specifications for procurement of the machine. Now this is the, you can see this is the machine BD, and this is the consumable required for bottle which is required to be purchased commercially available. After doing the barcode scanning, it will show the slot where we should put that bottle, and it will continuously provide the necessary environment, temperature, and in case if there is a growth, it will display in which slot there is a growth, and we have to take out and go for the further processing. So this is the BACT alert machine. This screen is there, and this is the cabinet where we can uh, load the bottles. So this is the consumable bottle. So anaerobic bottles are available, pediatric bottles are available, then adult blood culture bottles are available. So these two systems, BD and BACT alert. So these are the commercially available, very popular brands for blood culture system. So this is the Vitek, which is automated ID and AST. So different cards are available, cassettes are available. Like old days videos, this CD were there, audio cassettes were there like that. So it is the microbial pure inoculum should be inoculated into the this card, and it is loaded in the machine, and it will show the identification. And similarly for antimicrobial susceptibility also, similar cassette is available. So we have to purchase it separately depending upon the organism, whether it is a gram positive, gram negative, and it will show the antimicrobial susceptibility pattern. So for one-time investment is the equipment. It may go up to the 20 lakhs or even above depending upon the configuration. And then the repeated cost, recurrent cost is there of these consumables. So these are various machines. Now come to the automated systems for mycobacterial ID. So first, Bactec 460 TB, which utilizes radiometric principle, Middlebrook 7H9 broth or 7H12 broth, which is with bovine serum, albumin, panta, and catalase. So these are the ingredients of that, and turnaround time 14.8 days. Then Midget Mycobacteria growth indicator tube, Midget 960, which utilizes fluorescent mechanism for detection. The turnaround time is 13.3 days. Then Mycobacteria BACT alert system, which utilizes calorimetric principle for detection. The media use this Middlebrook 7H9 broth in CO2 and oxygen and nitrogen under vacuum, and supplementary ingredient is Panta. So turnaround time is 16 days. Then Bactec Myco F lytic. So this is a fluorescent based technique. It is the turnaround type is 12.8 days. So F lytic, the name, if you see in this system, something interesting is there, where the patient specimen is subjected to lysis, and the mycobacteria which are trapped inside the leukocyte, it is taken out. So to some extent there is an improvement and detection little bit earlier. You can see 12.8 days. So previous you can see 14.8, 13.3. So that lytic system is there. So lysis of the leukocyte and mycobacteria which are entrapped inside the leukocytes are taken out and cultured. Then gene expert CBNAT. So CBNAT, all of you know, it is a molecular method. The turnaround time is within 2 hours. But totally it is a molecular method. We are detecting RPOB gene. Then molecular automation. So this is all what we completed till now. These are the automated blood culture and automated AST. Now come to molecular automation, fully automated and consolidated into single system. So it is consolidated single system. The sample to answer, upload the sample and we'll get the result. Walk away. Just load the instrument, go for some other activity, and afterwards you come back, revisit the equipment. It will show the answer what is the organism isolated. So that is a molecular automation. Then total laboratory automation, TLA. So it is evolving quickly. It is evolving quickly. Where to pick up the colony, charging the panels, then preparation of the dilutions for susceptibility. So this is there and currently provided by Biomerix, BD, Kestra, and Copen. So these all these things, manual things are done by these automated equipments. Now come to advantages and disadvantages of automation. Advantages, of course, we are spending so much money for investment. So it is a labor saving, reproducibility, same result will be obtained. There is expert analysis and rapid results will be obtained. So turnaround time is decreased. AST can also be performed. What are the disadvantages? High cost. AST panels are predetermined. So suppose if you want to do something changes in the AST panel, if you want to customize the AST panel, it's not possible. Pure growth is required. A little bit contamination will also, all the things what are we are utilized will be waste. The inability to cover all organisms, whatever the spectrum of organisms or those cassettes are given, that only we have to utilize. We can't go for cover all organisms. Not able to detect the resistant phenotypes. So these are the disadvantages. So we have to balance what are the advantages and disadvantages, and depending upon that, we have to select what method is to be used. Now come to the next point, that is the proteomic based automated identification system. So proteomic, as such the word, what do you mean by proteomic? Proteomic is the study of interactions, function, composition, and structures of proteins and their cellular activities. Proteomics provides better understanding of the structure and function of organism than genomics. So this nowadays it has become very popular. Matrix assisted laser desorption ionization, time of flight. And what is this MS? Mass spectrometry. So here the two things will happen, desorption and ionization occur with the help of energy from laser. Desorption and ionization occur with the help of energy from laser. So here, what is the principle? Now we have to start from this line I will put. So if there is some culture in our lab, so it is put in the MALDI plate. You can see the culture, a single colony is taken and it is overlaid with a matrix. So what is matrix? Matrix is the mixture of the chemicals which will facilitate the ionization of the molecules. So this is the overlaid plate and this is loaded in the MALDI TOF machine and it is exposed to the lasers. So with the help of laser energy, these ions will be passing through the tube through positively charged electrostatic field. So laser assisted ionization will occur and from the laser exposure of laser to the that MALDI TOF plate will release the ions and these ions will pass through the time of flight tube, which is a vacuum tube, and the ions are separated according to mass charge ratio and it will go through the time of flight tube and into the ion detector machine, where machine compares with the database. So there is a formation of spectrum. So this is the spectrum, formation of spectrum, and the machine will automatically compare with the database available with it and gives the analysis of the identification of the organism. So this is the principle of MALDI TOF. Matrix assisted. Matrix assisted means this blue color what we have seen. Matrix is the mixture of certain chemicals which will help ionization and desorption of ions. So matrix assisted laser desorption. So laser is helpful for desorption. Desorption is something opposite of absorption. Absorption, they will absorb on the surface. Desorption means they will get released from the surface. So ion gets released because of laser energy and go to the time of flight, and then it is detected. So what do you mean by time of flight? Depending upon the mass charge ratio, the heavy molecule will go slowly, light molecule will go rapidly. So which will produce the spectrum on the detector system. So the smaller molecules are very active. You can see the lean and thin person can run very fast. Heavy and obese person will go slowly. So this is the mechanism of MALDI TOF. So you can see this. You can see sample matrix is there here, and with the help of laser, there is a release of ions, and the ions will pass through electric field generator, and then further they will pass through the time of flight tube, and afterwards they will go to the detection, which will produce the spectrum, and that spectrum is analyzed by the machine. So this is the machine, MALDI TOF, very costly equipment, maybe 1.5 crore. So it is in crores. So what are the advantages of MALDI TOF? MS, minimal sample preparation. It is a cost effective. So cost effective means initial investment is very high. I told you in crores, but consumables, it seems they are a little bit cheaper. Then powerful bioinformatic approaches are used. Then species to strain resolution is also very nice. Non-expert identification is possible. The day may come where just the technician may able to do the reporting, and we just sit on the chair and sign the reports. Then dedicated databases continue to expand. So this is the now in the beginning phase. So over a period of time, databases will get enriched. So again, it will improve the quality. Turnaround time is very less. So again, it is beneficial for the patient, especially critically ill patients, for management of critically ill patients, then for prevention of antimicrobial therapy, prevention of antimicrobial drug resistance, sorry. Then single colony is required. And for blood culture, because usually single growth will be there, we can inoculate directly in MALDI TOF machine. There is a low exposure risk. Everything is happening inside that machine. So the technicians or the laboratory worker will not be exposed to the infectious agent, and broad applicability. Anaerobes, we can detect yeasts, then filamentous fungi can be detected by MALDI TOF. I have noticed that many students, they will write viral diagnosis by MALDI TOF, but it is not correct. It is not applicable for diagnosis of viral infections. So you can mention in bacteriology, anaerobes, all bacteria, yeasts, and even fungi. Mycology also it is helpful. What are the limitations of MALDI TOF? Polymicrobial. Again, if there is a many organisms are there, it will get jumbled up and the results is not clear. So always at most care should be taken so that our growth is a pure growth. Some organisms require extraction, then pure culture is required. Databases are still developing phase, and there is a high initial investment. So a lot of money is required for its installation. Recently, I inquired for MALDI TOF for one test, 10,000 rupees cost is there. So you can see high initial, so investment is there, so they will charge more. Then criteria for evaluation. Now total so many automated methods we have seen. Automated blood culture we have seen, automated AST, ID we have seen. So during selection, we should select it properly. So we have to see the productivity how much are the samples coming to our lab and how it can be utilized, because we are spending so much money on those equipments. The reliability and durability, stability, we have to see whether there is a post that once we purchased it, post marketing or post purchasing, how much servicing they can give. The infrastructure, whether the related infrastructure is available. Some equipments require AC, continuous AC supplies required for maintaining the temperature. So all those things we have to take into the consideration. Only purchasing the equipment and keeping in some substandard level is not correct. Then software application, what are the softwares required? Sometimes we may purchase the equipment, but it will not cover, the software will not be covered. So we have to study it properly before giving the intent for the equipment. Then we have to see the biosafety levels, then QC and scientific aspects of equipments. There will be database. We can upload our results directly. For example, WHO net, WHO net is there, where we can directly upload the data of antimicrobial drug resistance. So such facilities there or not, we have to see. Then cost. So all these criteria we have to take into the consideration while picking up any equipment for procurement. Now, the last slide here, that is concluding remarks. The day may come, the microbiologist have to just sit in the chamber and report it. So much standardized test may come in future. Just we have to see the computer screen or your mobile screen and give the report. It is quite possible. But however, we should not forget the conventional methods. So conventional methods like microscopic method or culture identification by biochemical methods are also very important, because these are the basics and we have to use balanced approach. So wherever possible, the we have to utilize the conventional methods, and if it is not possible, we have to go for the automated methods. So this was all about the automated methods. So PG students, there will be essay question for you in the exam. So I request you, all of you, prepare this topic properly. Thank you for your patient listening. Thank you.